everyone. Thank you so much for coming today. So we are honored to have Dr. Alain Dagher today. And so he'll be talking about using computational models and genetics to infer causal mechanisms from big neuroimaging data. And so just a short introduction. He is a neurologist and a neuroscientist at the Montreal Neurological Institute and Hospital and McGill University, whose research focuses on the function of basal ganglia and the role of dopamine in cognition, emotion, and motivated behavior. His work investigates how we learn about reward and punishments and become motivated to engage in reward seeking behavior, with particular emphasis on Parkinson's disease, stress, drug addiction, pathological gambling, and obesity. Using advanced neuroimaging techniques like PET and fMRI, his research examines how disruption in dopamine functioning contributes to motor diseases and addictive diseases and explores behavior, endocrine, and metabolic correlates of these conditions. He was the first to publish on functional brain mapping in Parkinson's disease and has developed methods to measure dopamine release in the human brain, and also works on important insights into neural mechanisms underlying reward processing and disease vulnerability. So I would like to welcome Dr. Alain Dagher to our CIC lectures.
Okay. Thank you very much for the introduction, and thanks for attending. So I'm going to talk about a very specific issue. Many of us use big data, large-scale data sets that are now available or have become available recently. And we want to use these to infer causal mechanisms. And that's not always obvious how we can do that. So before I start, I want to thank all the students. I'll mention them later when I show their data. And especially my very close collaborators, Bratislav Misik, who is a collaborator on almost every study that I do, Yashar, who was my PhD student and continues to be a collaborator, and the other individuals mentioned here. So when we have large MRI phenotype data sets, we want to use them to understand mechanisms, carry out subtyping in diseases, develop biomarkers. And these are most of our analyses are based on identifying correlations between brain and certain phenotypes. However, as everybody knows, correlation does not equal causation. However, correlation does imply some sort of causation somewhere. And I've been very much inspired by the work of Judea Pearl in this book, The Book of Why, which I highly recommend, because he is one of the main scientists who's developed this new science of causal inference from observational data. And in this book, he explains all the ways that you can move from simple observation and correlation to causal mechanisms. Two of the things he proposes to do that are the use of causal diagrams to try to understand the organization of interactions, and then to use symbolic language to express what we want to know, and specifically to use biologically informed models. And he has this ladder of causation. This is taken from the book, this figure. I don't really understand all of the diagrams here, but he says that there are 3 levels of causal inference. The first is seeing or observing associations in the data, which we all do. This is what everybody does, use statistics to find patterns in data. The next level would be doing, so an intervention, so like a randomized controlled trial. If you think that hypertension causes stroke, one way to prove that is to treat hypertension and see if that reduces the incidence of stroke. But then the last and top level is imagining and using counterfactuals, that is, developing a model or an experiment and then changing the parameters of the experiment to see how that changes the outcomes. So I'm going to show you examples from some of the studies we've published recently that are inspired by these 3 levels of causal inference. So the problem with inferring causality from data, one of the main you all know about this, is confounders. So you find an association between A and B, an exposure and an outcome, an MRI pattern and a phenotype. And you want to know if it's causal, if A causes B. And one way that A doesn't cause B is if there's a confounder. One of the most famous confounders is the effect of red wine on health. There have been multiple studies showing that red wine is beneficial for health. And it turns out that it's not a direct effect of wine, unfortunately. It's actually a confound, and usually the most common confound is socioeconomic status. So people who are wealthier are more likely to drink wine, and people who are wealthier are more likely to be healthy. There are other confounds as well. The good news, though, is that moderate wine drinking is probably not bad for your health also. Another famous example of a proposed confounder is personality. So someone proposed that cigarette smoking did not cause cancer. This was in the 60s and 70s. And he proposed that it was actually personality, the type A personality, that caused you to smoke and also caused you to have cancer. Does anyone know who that person was? Who made this? No, it was H.J. Eysenck, who was a personality psychologist and actually the father of personality psychology, very, very prominent researcher. And he ruined his reputation by making this, and he took a lot of money from big tobacco to make these arguments. So, of course, you have to be careful, and when you account for confounders, and you also have to be careful with something called a collider. So a collider is also related to your two phenotypes of interest, but in the other direction. So a collider would be a measure or a phenotype of some sort that's caused by both the exposure and the outcome. And you don't want to correct for the confounder, the collider, because it has no way of causing an incorrect association between A and B. And this is actually very important in the kind of data that we study. I'm going to give you a cute example of a collider. And one of the problems with colliders is if entry into your study is predicated on the collider. This is a really kind of cute example of this. This is a map of actors' attractiveness versus acting ability. And so the question is, are actors who are more attractive worse actors? And if you look at the entire data set, this is simulated data, but if you look at the entire data set, there's no correlation. But if you were only to look at Hollywood actors, you would find that they're all in red. All the Hollywood actors are in red. So they're either very good looking or they're good actors, or usually both. And when you look for a correlation between these two, you find that there's an inverse correlation. So in fact, you've shown that good-looking actors are worse actors, but it's a collider bias, because entry into your new study, being a Hollywood actor, is predicated on both acting ability and attractiveness. The really interesting thing is that if you did the correlation amongst non-Hollywood actors, you would also find a negative correlation. That's called Simpson's paradox, where you have a correlation in one part of your sample and in the other part of your sample, but in fact, there is no real correlation. It's not just cute examples like this. A prominent example of collider bias in the kind of science we do is attrition bias. So when you do a study on Parkinson's disease or Alzheimer's disease and you follow people over time, people will tend to drop out of the study who have worse disease, who have more severe disease. So that's a collider, because now entry into your study is based on having a milder form of the disease. So when you look for association between disease severity and some measure, you're prone to collider bias. And this is actually something that has been observed in the PPMI data set for Parkinson's disease. All right, so now I'm going to show you some ways that we can get around these various problems and still infer causal mechanisms in big data. So I'll start with this study that was done by Mari Shishikura, a PhD student in my lab, and who was interested in impulsivity. I'm sure most of you know what impulsivity is. And there have been many studies showing that impulsivity is related to the dorsolateral prefrontal cortex being involved in executive control or self-control, and ascending catecholaminergic systems involved in drive and motivation. But most of the studies in the literature are prone to small samples and also to the confound of the fact that if you're impulsive, you are going to be exposed to things that can change your brain. So if you're impulsive, you're more likely to smoke, drink alcohol, you're more likely to have stress, and all those things will change your brain. So when you see impulsivity brain associations, you can't be sure that they're causal in the impulsive phenotype. So Marie's idea was to use genetics, and that's one of the themes of this talk, is to use a third measure to try to infer causality. And here we were very fortunate that collaborators in San Diego, Sandra Sanchez-Roig and her team, recently published multiple GWASs of various different forms of measures of impulsivity. So you can measure impulsivity by self-reports. These are validated scales of impulsivity, by tasks like delay discounting, and by life events like smoking, drinks, automobile speeding, and things like that. And then you can have related phenotypes like obesity and educational attainment. So recently, people have published GWASs of all of these 17 different phenotypes. And what Marie did was to use something called genomic structural equation model to try to disentangle causal mechanisms in impulsivity, and specifically to look at genetic mechanisms. We know that impulsivity is highly heritable, and so there must be a genetic cause to it. So genetic SEM is just like structural equation modeling, but using GWASs as your main measure. And what you can see, so this is a structural equation model with latent factors. You can see that in the genetics, we identify 3 different latent factors of impulsivity. Now, this is entirely driven by genetics. And we labeled the 3 factors which are related to the different measures. We label them lack of self-control, reward drive, and sensation seeking. And what's interesting here is that this distinction between self-control and reward drive is also seen in the behavioral phenotypes. This is something that comes up in human and animal research as being 2 factors, 2 independent factors contributing to impulsivity. So the genetics also confirms this split. What you can then do, which Marie did, is you can take these latent factors and actually compute GWASs for these latent factors. Even though you can't actually measure this, you can't measure F1 in a person, but you can infer a GWAS based on the genetics of all of these other factors. So we were able to do these 3 latent factor GWASs for the 3 different impulsivity phenotypes, which you can see here. The other thing that is interesting that you can do, you can use the BrainSpan data set, which has sampled gene expression at different phases of the lifespan from the fetal stage all the way to old age. And what Marie was able to show was that the reward drive phenotype, latent factor, was related to genes expressed in the early prenatal phase, whereas the sensation-seeking latent factor was more related to genes expressed during late infancy and adolescence, so suggesting different influences on brain development. And then the other thing she was able to do, you can read her paper, is then relate each of these latent traits to brain. And for this, she used the ABCD cohort of, in this case, about 4,000 people, children aged 9 to 11. And without going into details, she was able to show that the different phenotypes, the different latent phenotypes, were associated with different brain abnormalities. So the lack of self-control latent factor was associated with cortical thinning and reduced surface area, especially in prefrontal areas, which fits with what we know about the mechanisms of self-control. Whereas the reward drive phenotype was most associated with subcortical measures, again, fitting with what we know about the reward drive mechanism. Moving on to another condition and another way that we can use genetics to try to understand mechanisms. This is a study on essential tremor done in collaboration with Guillot, and the 2 people who did the work are Miranda Medeiros and Alex Pastor. So essential tremor is a movement disorder. It's actually the commonest movement disorder, characterized by action tremor. So this is a sinusoidal involuntary movement of the limbs during voluntary action, and it can be quite debilitating. So essential tremor can start anytime between the age of 20 and 70. It responds to deep brain stimulation of an area of the thalamus called the VIM, and it's also heritable. There's a strong familial component. Actually, 50% of people with essential tremor have a family history. And there was a recent GWAS performed by Giroulo's lab, which showed actually quite high SNP heritability of 20%. So, normally SNP heritability is much lower than twin heritability, and 20% is quite high. So that means that the GWAS explains quite a bit of the phenotype. So what we're interested in is trying to understand the brain mechanisms of essential tremor. There are very few published studies on essential tremor with MRI, tiny sample sizes, very inconsistent findings. But in this case, we were able to use the UK Biobank with 30,000 individuals who had brain imaging. We excluded people with essential tremor or a family history of essential tremor, and we assessed the relationship between brain MRI measures and polygenic risk score. So here we're taking people who don't have the disease, but who score high on a genetic risk factor of the disease. And we're asking what in their brain may be different that makes them vulnerable to the condition. And in this case, we specifically used diffusion-weighted MRI and cortical thickness measures from the UK Biobank. And what we found is that the essential tremor polygenic risk score was associated with widespread microstructural changes in white matter, but especially in a specific tract, the cerebellar or dentato-rubrothalamic tract, which is identified here, cerebellum, brainstem, red nucleus, and thalamus, and going all the way to motor cortex. So what we find is that in these white matter structures that connect the motor system to the cerebellum, we find that there's evidence of increased mean diffusivity, which is perhaps indicative of microstructural damage. And we know that this is the tract that's important for essential tremor because it's the tract that responds to therapy. So when people use deep brain stimulation or lesioning of the thalamus to treat essential tremor, you can look at the connectivity of the area that has the best effect on the symptoms, and this is what comes out. This is the connectome of the therapeutic lesion in essential tremor. So in other words, we would suggest that people with high genetic risk for essential tremor have microstructural abnormalities in white matter tracts that are implicated in motor control. Now, of course, something else has to happen to tip the person into the actual disease phenotype, and we don't actually know what that is. We don't have any idea what that is. The other interesting finding here is that the other area that's affected is the orbitofrontal cortex. So connectivity of the orbitofrontal cortex is also affected. And that's really interesting because people with essential tremor have a higher incidence of anxiety disorders and alcohol use. And that's always attributed to the fact that alcohol treats the tremor. So that's another feature of essential tremor. It responds very well to alcohol, and people assume that's why there's an increased incidence of alcohol abuse. But it's conceivable that there's also some abnormality in this limbic system known to be implicated in substance use.
So is this a role for dopamine? Yeah.
No. So essential tremor, number one, doesn't respond to dopaminergic treatment, so it's different than the Parkinsonian tremor. And there's no genetic overlap between essential tremor and Parkinson's disease. So it's a different disease. The other thing I wanted to show also, we dove into the specific organization in the thalamus. And what you can see is that this VIM nucleus in the thalamus shows the greatest effect of increased MD related to genetic risk for essential tremor. And as I said, this is the target. This is the area where you lesion this target to reduce the tremor. So in conclusion, essential tremor genes, we think affect neurodevelopment. I didn't show that part, but there's evidence that these genes are actually expressed, especially during early prenatal life. So we think that these genes affect the brain development of motor control tracts and associated gray matter nuclei, and that some unknown environmental trigger may therefore cause further white matter injury, bringing out the phenotype.
So as a UK Biobank is a longitudinal study, there's then a great opportunity as
absolutely
to prove the point.
Absolutely. So in UK Biobank, there are some people with essential tremor. This is actually in the paper, I didn't mention it. So we looked at the people with essential tremor, and they have white matter abnormalities in these tracts as well. The other thing is, yes, as you said, it's a longitudinal study both clinically and with imaging. So it will be very interesting to study this. So now I'm just going to move on to Parkinson's disease and to two studies that we've done to try to understand causal mechanisms in Parkinson's disease. I'm going to try to relate them to Judea Pearl's third level of causal inference, the counterfactual imagination approach. So this is a paper by Stan Prusner, the person who discovered prions, in the New England Journal of Medicine. He said, 25 years ago, little was known about the cause of neurodegenerative diseases. Now, however, it is clear that they result from abnormalities in the processing of proteins, namely prion diseases. In each of these diseases, defective processing causes the accumulation of one or more specific neuronal proteins. And I think now everybody would agree. 10 years ago, most people didn't agree, but now most people would agree that this abnormal proteostasis is the cause of pretty much all of the neurodegenerative diseases. But we have been interested in my lab in trying to model Parkinson's disease to try to understand these mechanisms. So the first person who proposed that Parkinson's disease was a stereotyped progressive propagating illness was Braque. And he did this based on his observations of postmortem samples. So he looked at postmortem samples and looked for the specific pathology of Parkinson's disease, which is called Lewy pathology. And he found that there was a stereotyped organization of spread of the disease from the medulla, and specifically the dorsal motor nucleus of the vagus, to the upper brainstem, to the amygdala, and to the cortex. And he described 6 stages of this stereotyped propagating condition coming from the medulla up into the cortex. And he inferred that the disease may be caused by an environmental toxin, because the 2 parts of the brain that were most affected early on were the olfactory bulb and the dorsal motor nucleus of the vagus. The olfactory bulb is connected to the outside world, and the vagus is connected to the gut. So if there was some sort of agent in the environment, it would go via the nose and the gut. So that was his theory, and I think he may well be at least partly correct. Interestingly, Braque also had cases of Lewy pathology in the limbic system, but not the brainstem. And he actually discarded those cases. So it's kind of interesting because obviously you shouldn't be allowed to do that. But despite the fact that he did that, I think he was still mostly right. He was only sort of 80% right. We now think that Parkinson's disease, or at least Per Borghammer has suggested that Parkinson's disease actually has 2 propagation routes at least, one from the gut to the brain, the so-called body first, and one that starts in the brain and moves downwards, the so-called brain first. So there have been studies on brain atrophy patterns in Parkinson's disease, but until pretty recently, they were all based on very small sample sizes. So in my lab, we've been trying to do large N studies of Parkinson's disease atrophy. And this is probably the largest study that we've done to date. It's the work of Andrew Vo with the Enigma Consortium. So he was able to get data from 3,000 patients and 1,000 plus controls from the Enigma Consortium. And he found that there was a quite prominent pattern of brain atrophy in Parkinson's disease. You can see here cortical thinning, so widespread cortical thinning and reduction in cortical surface area, and also widespread reductions in volumes of all the subcortical nuclei. So Parkinson's disease, even though it's thought to be a motor syndrome that affects the substantia nigra, actually affects the entire central nervous system, even early on in the disease. So, he looked at people within Enigma with stage 1 disease, the earliest disease stage, and also already found evidence even of cortical atrophy. So the idea being that the disease, by the time you present with symptoms, the disease has spread to affect the entire nervous system. And the other thing that Andrew did, and is something that we routinely do in the lab, is to ask whether the pattern of atrophy could be explained by the connectome. Because if, as Prusner suggested and Brack suggested, Parkinson's pathology spreads through neuronal connections, something that everybody accepts now, but if that's the case, then the atrophy of any brain region will be explainable by the atrophy of its connected neighbors. And so this is just a way to ask this question. So you can measure the atrophy for every node, you can measure the atrophy of all its neighbors and see if it correlates with the atrophy of that node, after correcting for age and sex and disease stage and all those things. And in fact, this is what we find. We find that there's a very strong effect of connectivity, whether you use a structural connectome or a functional connectome. So consistent with the idea that that the connectome is important in shaping the pattern of atrophy. Of course, correlation doesn't exist, it doesn't equal causation. Two connected areas could show coatrophy for a whole bunch of reasons. For example, in the brain, areas that are connected tend to be similar in many ways. They tend to have similar gene expression patterns. They tend to have very similar cytoarchitecture. So it's quite possible that coatrophy doesn't necessarily prove a propagating phenomenon. So how can we use Judea Pearl's third level of imagination to try to infer this causal propagation mechanism? So one way that we came up with was to use a biological model of the disease and to do manipulations on the model instead of manipulations on patients. And so I mentioned the prion hypothesis, but this shows it in more detail. So the idea is that alpha-synuclein can become misfolded, it can aggregate, it can spread from neuron to neuron. In its misfolded state, it tends to aggregate and is neurotoxic. It damages brain cells, mitochondria, lysosomes. And the other important feature is that a misfolded protein can misfold other proteins by a chaperoning effect. This is how prions work. So the misfolded protein meets a normal protein and causes it to misfold. And so we wanted to test this theory, but before I show this, I want to do a little side trip into how you can make inferences about mechanisms from observing spatial patterns. And I used to show snow and the cholera epidemic in London, but actually now I find this even more interesting. This was given to me actually by Albert Getty, a friend and mentor. So Panem, for those of you who've been to Copenhagen, there's a Panem Institute. It's named after Peter Panem, who as a young medical student or young doctor, I think it was 22, was sent to the Faroe Islands. The Faroe Islands belong to Denmark, and there are these islands in the North Atlantic that have a very peculiar organization in that there are all these villages, these fishing villages around the edge of the islands, but there are no roads between the villages. So there's very little communication from village to village. And Panem was sent there to study the measles epidemic. So there was a measles epidemic in 1848. And in 1848, people still didn't know what caused infectious diseases. It was still controversial whether a person could infect another person. And what Panem observed, he made many observations, but one of them is that the disease would spread in one village and everybody in the village would get measles. And only when a person traveled from one village to another village would the disease spread to that village. Even if the village was very close physically, there needed to be someone who went by boat from one village to the other. And so that really supported the idea of transmission from person to person. The other thing he observed, which is really interesting, is that the epidemics in the Faroe Islands recurred on 65-year cycles. And the reason for that, he correctly realized, is that measles gives you lifelong immunity. And only when there are enough people who are vulnerable or susceptible to measles could the epidemic be triggered again. You didn't have epidemics on the mainland because there was way more travel. So there was constant level of measles. But in the Faroe Islands, it came in these 65-year cycles of massive epidemics. So, very interesting how you can infer a mechanism just from observing the spatio-temporal distribution of the disease and knowing something about the anatomy or the geography of the area. So what we're going to do.
But this is actually interesting because it could affect young and older and middle age. So it's all synchronized at 60, 65. Very interesting.
Because you have to have a lot of people who are susceptible. Yeah. And it's herd immunity is basically, we've heard about this with COVID. That's what it is.
But they need also be similar age, generation-wise synchronized.
Yeah.
Cool.
So now there are people who are using much more sophisticated methods to study epidemics. And this is the work of Brockman, which was actually pointed out to us by Yasser Ituria Medina, who many of you know. So they have been using models of infectious diseases that spread through the entire planet using airplane routes. And what they've shown in several studies is that the best way to model the spread of infections like influenza through the world is by using air travel. So for example, if you look at the spread of this simulated, I think this is influenza, you can see here he's plotting the arrival time of the disease. It starts in one epicenter and spreads through the entire planet. And you can see that the arrival time of the disease is not correlated to the physical distance between cities. However, if you use effective distance based on the connectome, in this case, airplane travel, you see that there is actually a correlation between effective distance and arrival time, proving that the disease is spreading from city to city by airplane travel. And then he did this for COVID, many really fascinating papers. This is showing all the new COVID, the different subtypes of COVID, where he was able to plot the origin, the epicenter, and the arrival time, and show that for every single new variant of COVID, the arrival time in any city was based on the airplane travel. And the reason this is interesting to do these kinds of models is that then you can do interventions. You can ask, what will happen if we don't let anybody board a plane who has a fever? And so you can actually test interventions without testing them, without having to wait 10 years for the outcome of a study. So we were inspired to do something like this. And this is the work of Yingqiu Zheng. She disappeared. She was on the previous picture. Or maybe. So instead of cities, we're going to have brain areas. Instead of people, we're going to have prions or alpha-synuclein molecules. And instead of airplane travel, we have the connectome. And so this is called a susceptible, infected, removed agent-based model, where each protein can exist in one of three states: a susceptible state, the normal state, an infected state where it can infect other proteins, and a removed state where it's metabolized and cleared. And then in this model, when enough proteins accumulate in a neuron or in a brain area, we assume that leads to cell death and therefore brain atrophy. There's Yingqiu. So she did this model, and so used a susceptible, infected, removed model where the prion moves from neuron to neuron, used the connectome, a structural connectome, as the the backbone of propagation of the disease. And the other thing that she did is to emphasize that this was alpha-synuclein. She used two genes, GBA, which is a gene that's involved in clearance of alpha-synuclein, and mutations in GBA cause Parkinson's disease, and SNCA, which is the alpha-synuclein gene, also a gene where mutations can cause Parkinson's disease. So in this model, you have the connectome. The synthesis of alpha-synuclein is determined by gene expression of SNCA and the clearance by expression of GBA. And then, so we used the gene expression of GBA and SNCA from the Alain Human Brain Atlas. So we have the actual expression of each gene throughout the brain. We have a connectome from diffusion MRI studies. And we ask, does the spread of alpha-synuclein in the model replicate brain atrophy? This is from Yashar's paper, 10 years old already. This is the atrophy pattern in Parkinson's disease. So we let the model run and we ask, does accumulation of abnormal alpha-synuclein in this model reflect brain atrophy? And indeed it does. So when you look at simulated atrophy versus true measured atrophy, you see a very strong correlation. So in the model replicates the atrophy pattern. Now that in itself is not interesting. The reason to do this is to do that third level of inference, to then say, okay, if we disrupt the model, can we change the fit? And if that's the case, then the thing we disrupted was important for the disease. And so this is what Ying-Chu did. This was mostly her work and Bratislav's ideas. So for example, if we take the SNCA gene, so this is the true distribution of SNCA in the human brain. Interestingly, it's most highly expressed in dopamine neurons. But if we take the SNCA gene expression and then randomize it and rerun the model, what happens? And so this is the model in blue, rerun, I think 5,000 or 10,000 times. And this is the distribution of correlations between the model and the true atrophy pattern. And what you can see is that when you use the real SNCA value, you have a much more significant fit. So what this shows is that when you disrupt this part of the model doesn't fit anymore, supporting the idea that SNCA was important and that what's happening is propagation of alpha-synuclein. She also did this for GBA and the same thing. So this is with GBA. So in gray are all the null models and in red is the true model. So you can see that the fit is much better with GBA, supporting the idea that clearance of alpha-synuclein is important for the disease propagation. She also did the same thing with the connectome. So shuffling the connectome with either spatial null or rewired nulls, and the same thing. When you disrupt the connectome, you reduce the fit. So altogether, this says two things. This counterfactual model says that alpha-synuclein is probably the toxic agent in Parkinson's disease, and it does spread through the connectome. So, and then the last thing, so that's one area of research. The next question, though, is it now seems to be the case that alpha-synuclein misfolding is something that happens commonly in humans. Postmortem studies, there's a lot of evidence that alpha-synuclein can misfold spontaneously, probably triggered by inflammation, maybe by toxins, but not everybody gets Parkinson's disease. In fact, most people don't get Parkinson's disease. So the next question is, what is it that makes a person or a brain vulnerable to synucleinopathy? And so part of this is asking what makes people at risk of developing the disease. And there are known behavioral and demographic phenotypes in PD. So for example, cigarette smoking, surprisingly, is associated with a lower risk of Parkinson's disease, and I'll get back to that. And educational attainment is associated with a higher risk of developing Parkinson's disease. In fact, educational attainment, socioeconomic status, and IQ are all associated with a higher risk of PD. It's the only disease, as far as I know, maybe obsessive compulsive disorder also, but this is really one of the only diseases I know that is more frequent if you have higher education and higher socioeconomic status. And I think we know why, as I will show you now. So there is a GWAS of Parkinson's disease. So Parkinson's disease, like most neurodegenerative disease, there are rare familial cases, less than 10%. But in the non-familial, so-called sporadic cases, there's also a genetic risk. And it is somewhat familial. And there's recent GWAS of PD. In fact, actually, there's a bigger one now, but based on something like close to 40,000 cases, 18,000 proxy cases in UK Biobank. So those are first-degree relatives, and then 1.5 million controls. So very strong GWASs. If you have high polygenic risk for PD, so this is the polygenic risk score quantile, and this is your risk of developing Parkinson's disease, you can see that it goes about an 8-fold increase in your risk of PD if you are in the top quartile. That still only means you have an 8% risk of developing the disease at age 65. So even if you're a very high genetic risk, you probably won't get Parkinson's disease. So this is work that was started by Nushin Abbasi and then has been taken over expertly by Human Azizi. So similar to the essential tremor study I showed you, here we asked, is the Parkinson's disease polygenic risk associated with brain morphometry? And our initial hypothesis was we'd find the same thing, that we'd see that people at high risk for PD would have subtle subclinical evidence of Parkinson's disease. So in about 30,000 people in the UK Biobank, we did the same thing, compared polygenic risk score to brain measures. What we found is the opposite of what we hypothesized. So the first finding is that high genetic risk of Parkinson's disease is associated with increased cortical surface area in the entire brain. High polygenic risk, there's also an effect on cortical thinning, so reduced cortical thickness, but it's very scattered and very, very mild. So the main finding is really this increased cortical surface area in people at high risk for Parkinson's disease. So if you remember what I told you about IQ and education, maybe it's starting to make sense. There's also an increase in subcortical volumes in Parkinson's disease. Now remember, people with PD have smaller subcortical volumes, but people at genetic risk who don't have the disease have increased subcortical volumes. And then finally, we see increased fractional anisotropy in the entire white matter, in all of the white matter. So in other words, increased cortical surface area, increased white matter density, and increased subcortical volumes. In other words, bigger brains with more neurons and more connections. And this is basically explainable by Rakic's theory, the radial unit hypothesis. And in that theory, cortical surface area is something that develops very early on in fetal development due to these neuronal stem cells, which divide in the ventricular zone and generate cortical units. And the longer they divide, the more cortical units are generated and the greater the cortical surface area. So you have greater cortical surface area and more neuronal projections based on genes that act before birth. So we would propose, based on this, that it's very likely that the genetics of the genes that cause risk for Parkinson's disease are doing it by increasing brain size, and they would be doing this early on in life. The other thing that Human found is that the polygenic risk score for Parkinson's, and now this is in, there's a typo, over 200,000 participants in UK Biobank. You can see that there's an increased educational level, increased household income, increased fluid intelligence, not a great measure in UK Biobank, decreased BMI, decreased multiple deprivation index. So what that means is that people at high risk for Parkinson's tend to live in neighborhoods that are wealthier. Decreased smoking, very strong effect on smoking. So to get back to smoking, Smoking is very rare in people with Parkinson's disease, and that's been attributed to possible neuroprotective effect. But it's quite possible, so these people don't have Parkinson's disease. It's quite possible that this reflects the known inverse association between intelligence and smoking in the current era. The other way to test whether an association is causal is something called Mendelian randomization. So we've seen this association between Parkinson's disease genetic risk and brain, but what causes what? So I won't go into too many details, but Mendelian randomization is a way to use genetics to find causal associations. And just because of time, I just will show the results. I'm sure you can look up Mendelian randomization. It's really quite fascinating. What Human found was that genetic risk for a bigger brain was causal on Parkinson's disease, but that genetic risk for Parkinson's disease was not causal on having a bigger brain. So once again, supporting the theory that genes that cause high risk for PD are doing it via brain size. We also found, Human also found that the surface area expansion map correlated with the cortical thinning map in Enigma. In other words, the brain areas that show the greatest surface expansion also show the greatest cortical thinning in PD, suggesting that this is, again, a vulnerability phenotype. So I won't talk about the pathway-specific polygenic risk score now because of time, but I'll just show this last figure. So I told you that genes that affect surface area are probably expressed in fetal life. And this is, again, using the BrainSpan data set showing. So these are the genes, the part of the polygenic risk score that we think is involved in cortical surface area. And what you can see is that these specific genes appear to be mostly expressed in early fetal life, and then their expression declines in childhood and adulthood, as opposed to these other genetic risk factors. But what this shows, consistent with all of the other mechanistic hypotheses, is that genetic risk for Parkinson's disease is lifelong. It's based on brain development, on effect of brain development that occurs before birth. So as I showed, genetic risk for Parkinson's disease is associated with increased cortical surface area, white matter density, and subcortical volumes, and related behavioral measures. Mendelian randomization suggests that cortical surface area expansion has a causal effect on PD risk. We did other parts in this study to show that these participants with high genetic risk don't have prodromal Parkinson's disease, both behaviorally and in the brain. And then, the other thing to remember, if that's the case, is that things like socioeconomic status, occupation, neighborhood, these are all potential confounds in epidemiological studies of PD. You may have seen a recent study showing that people who live near a golf course have a higher risk of Parkinson's disease. And so people have hypothesized that this is due to pesticides. Personally, this makes no sense to me. I don't think if you live near a golf course, your water is full of pesticides, but who knows? However, there's the confound that people at high risk for Parkinson's disease, as we've shown, tend to live in rich neighborhoods, including probably areas around golf courses. So the next question, the last question is that we're interested in is, why is that? Why is it that a bigger brain, so we've shown previously that Parkinson's disease and others have shown is due to propagation of this alpha-synuclein, misfolded alpha-synuclein, but why would a bigger brain be more vulnerable to this? So of course, one possibility is that a larger brain with more connections is just better at transmitting the prion. But another possibility is that a bigger brain has a lot of advantages, but it's also precarious energetically. And I think that's the explanation. I didn't mention this, but the cause of cell and synaptic death in Parkinson's disease is energy failure, is mitochondrial dysfunction. A lot of evidence of that. So it's conceivable that a larger, more connected brain is more energetically precarious, and that when it starts to fail, it fails faster. And the network theory for that is called cascading failure, first proposed by Duncan Watts of Watts and Strogatz fame. And the idea here is that certain very efficient systems are very vulnerable to assault, because as soon as one area fails, the neighboring areas now see greater stress and they start to fail. So that's something that maybe we could test at some point. So the cascading failure. Interestingly, in Alzheimer's disease, I don't know, maybe Sylvia can correct me, but I think people with higher educational attainment are protected against Alzheimer's disease, but when they get Alzheimer's disease, they progress faster. And there could be many reasons for that, but it could also be this, that bigger brain is more precarious and more vulnerable to cascading failure. So I will end there. Hopefully I've convinced you that we can take big data and not just look at correlations, but make inferences about mechanisms of disease. Thank you.
5 minutes per question. So if you would raise your hands here, but then very quick question is, so do you think that it's more in men or more at risk of Parkinson's? Do you think it's reasonable? No.
So that's a good question. So Parkinson's is more common in males, but the association, the genetic association with surface area was equally present in males and females. So I don't think that's the cause. I don't know the mechanism of relative protection in females.
I think maybe like having a bigger brain, evolutionarily speaking, like led people to have a more like turned off clearance system because it gives more space for the body to like store junk, for example. And then do you think there's like an evolutionary like balance between clearance and
Very interesting question. What I didn't mention, and it's not my area, but alpha-synuclein is actually involved in learning and memory and plasticity. I mean, its main role at the synapse is probably to regulate synaptic strength and pruning. And in fact, a really interesting finding, the alpha-synuclein equivalent in birds. So I don't know if you know songbirds, they have a critical period where they learn their song, very short period. And during that period, alpha-synuclein, or the equivalent, is way upregulated. So this may link alpha-synuclein actually to intelligence and learning. It may be a molecule that's very important for plasticity and learning. But as far as your question, so the whole clearance, garbage disposal aspect of Parkinson's disease is very interesting, but I don't know the answer to that question.
One final question, I mean, you addressed it in your own studies with looking at disease itself, but how worried should one be in these large data sets about the collider effect in the sense that they don't have the disease? So you're looking at risk factors, but maybe you're also looking at protective factors.
Mm-hmm.
How do you distinguish those without going into the actual?
That's a very good question. So obviously, in the UK Biobank, people at high genetic risk for PD, I told you they don't show Parkinson's disease, so they may be protected in some way. And yeah, I don't know. I don't even know how we would address that issue, perhaps by comparing, or by looking at the people who go on to develop Parkinson's disease. But yeah, that's a great question. I don't know yet. I don't think we know, anyone knows.
What about brain activity and people which are at risk? When you see bigger brain, is it also more active? Are they more alert when they have higher attainment levels, for instance?
Yeah, I don't know.
Function MRI data, anything on that?
Yeah, in Inkchu's paper, she actually showed that functional MRI, resting state fMRI, improved model fit as well. So there wasn't just connectivity, but activity also improved model fit. And alpha-synuclein is released in an activity-dependent manner. So when neurons fire, actually, consistent with its role in plasticity, alpha-synuclein is released by the presynaptic neuron and taken up by the postsynaptic neuron. So it's entirely possible.
Great talk. Thanks so much. So you talk about network efficiency in those regions, kind of the cascading effect of. So if you think of work that Steph's done or other work that like same thing as the work that inspired from your work. If we introduce alpha-synuclein, if there's too much alpha-synuclein in one part of the brain, the rest the regions that come next would be kind of a cascade based on network efficiency. Can you test that based on kind of network models what you already have? Like, you look at even the baseline network efficiency,
Mhm.
elderly adults, in those regions that would be most vulnerable, and is that support
Yeah.
the hypothesis?
So that's a case, that's true though for every neurodegenerative disease, that areas that are hubs, high degree nodes, are more vulnerable, right? Ed Bullmore showed that, and many people have found that. So I don't know what necessarily that means. One thing that will be interesting is when we have more longitudinal data in Parkinson's disease, it will be really interesting to test these kinds of questions. So for example, do people with higher cortical surface area who have PD, do they progress faster?
Yeah.
Or do those brain areas that are mostly affected by the genetics, do they progress faster? Or does resting state fMRI predict progression? Those will be interesting. I don't think we have the data yet in humans.
You can see it in conversion rates based on the MRI data to downstream conversion to PD, I think, like, so there's there's a scan that you have.
Yeah, in the UK Biobank,
Yeah.
there's not many people who have PD in UK Biobank though. Like and imaging, PD and imaging is very like 100.
Okay.
Thank you so much. This is so interesting. I was really interested in what you said in the potential environmental factors on PD because of the effects on olfactory system in the gut system. Is there like any proposed environmental factors or like any geographic data about people who are different?
Absolutely. And there's a new book actually that just came out by Ray Dorsey and Michael Okun, O-K-U-N, and Dorsey. I highly recommend. I don't know if they're right, but they've been proposing environmental factors in Parkinson's disease. They also have a blog, but they found links to pesticides, and there's a long history of pesticides in Parkinson's disease. In fact, in Quebec, some of the first research showing linked with pesticides was done in the 60s. And it's been shown over and over that people who drink well water, for example, are at high risk of Parkinson's. So there's this link with pesticides. There's also a link, believe it or not, with dry cleaning. You can look that up in Dorsey's book. The trichloroethylate or something is also toxic to neurons and can trigger synucleinopathy. So all kinds of theories related to environment. And I think there's good evidence that Parkinson's disease has an environmental cause. The other interesting thing is that it's one of the few diseases that's increasing in incidence, massively increasing in incidence. The incidence of PD is going to double between 2000 and 2050, which is quite scary to think of. And so people say, well, maybe it's because of some sort of toxic modern world product.
Like all the time we have for questions. Thank you so much, Dr. Dagher, for
Thank you.
giving us this insight.